Giuseppe Agapito and Mario Cannataro. The paper is called Using BioPAX-Parser to Annotate List of Biological Entities with Pathway Data. Please, Giuseppe, you can start.
You can see the slide. Okay, thank you. Hello to everyone. So I am Giuseppe Agapito, and this evening I present a work in conjunction with Mario Cannataro titled Using BioPAX-Parser BiP to Annotate List of Biological Entity with Pathway Data. The outline is the following. I start to give you a definition of a biological pathway and illustrate some of the most famous and used pathway database. After I introduce the pathway data formats used to represent pathway data, and I introduce the different type of pathway enrichment analysis. After that, I present BiP, a software tool that makes easy to perform pathway enrichment analysis by using pathway data coming from different biological pathway database coded in BioPAX format. And finally, I present a case study using BiP, and the conclude the paper. So, what is a biological pathway? A biological pathway represents the biological reaction and interaction network in a cell. Biological pathway can be grouped in three main categories, so metabolic, regulatory, and transduction pathway. It is now become very increasing and evident that the connection between pathways and disease is fascinated at the multiple interconnected level. So this means that when something goes wrong in a pathway, the result can be a disease, such as cancer or diabetes. Pathways have become the first choice for gain insight into the underlying biology of differential expressed gene and protein, as it reduces complexity and has increased the explanatory power. So, the very well-known pathway database include KEGG, Kyoto Encyclopedia of Gene and Genome, that is a collection of several databases. After, there is MetaSig, that is a curated database that contains information about metabolic pathway. After, we have PANTHER, protein analysis through evolutionary relationship classification system that include information about metabolics and signaling pathway, among the other available. After, there is Pathway Common, that is a collection of public databases that provide a common access to from a unique access point. After, there is Reactome, that is that contain pathway coming from 22 different organisms, including the human. And finally, there is Wikipathway, that is an open collaborative platform that try to improve and complement the effort of such database like KEGG, Reactome, and Pathway Common. Pathway are represented using different format. So, like BioPAX data exchange format, so BioPAX, that the aim of BioPAX is to define a unified framework for sharing the pathway information. After, another standard is PSI-MI, Proteomic Standard Initiative Molecular Interaction, that is a standard developed to represent proteomic and allow to compare, exchange, and verify data. And the other format is SBML, System Biology Markup Language, that is used to model biochemical reaction, including cell signaling, metabolic pathways, and gene regulation. After that, there is the use of plain text to represent pathway data, and even XML. The high level of heterogeneity among the available pathway makes automatic data analysis ineffective. Thus, an attempt to try to reduce this high level of heterogeneity is adopted by the BioPAX consortium by developing BioPAX languages. That is a meta language defined in OWL DL and represented using RDF XML format. So BioPAX purpose is to facilitate, simplify the integration and exchange of data maintained in a biological pathway database by using a unique data representation. Pathway enrichment analysis is very useful to gain information about the biological context of differential expressed genes. And pathway enrichment analysis, PIA, can be classified in three classes: overrepresented analysis, or gene set enrichment analysis, and topological enrichment analysis, TIA. So the first two categories of methods perform enrichment analysis using an input list of gene or protein or SNP along with a database of interest, whereas the third class add to the pathway enrichment computation the topology information of the network, if these are available. And here we have BioPAX-Parser, BiP, that is a novel software tool implemented in Java, able to perform pathway enrichment analysis using a list of gene or protein, performing enrichment by using a customized version of hypergeometric test, along with false discovery rate and Bonferroni corrector, used to adjust the p-value from error due to multiple statistical tests. BiP is very simple to use and can help user to extract information from each database is coded on BioPAX-Parser. At the moment, the current version of BiP can retrieve information from each database compliant with BioPAX format. Here there is a simple workflow of use of BiP. So from the main menu, you can choose the pathway enrichment analysis tab, set the information, choose pathway database and the input list of gene, and after BiP provide the enriched pathway by using that database and that list. And here there is the architecture, the summary of architecture. So the architecture of BiP is a modular architecture where BiP core is the main core that interacts with the user and acts as controller for the submodule. In particular, we have two different input reader, one compliant with BioPAX and another to read and translate the biological entity. So, and this is done in parallel by using Java multi-thread programming. After, to improve the pathway enrichment analysis computation, the RDF triple are converted in attribute and are indexed into the attribute container data structure. After, there is the PEA, that is a pathway enrichment analysis library, that is a statistical module that performs the statistical computation. And at the end, the graphical user interface visualize the result to the user. As case of study, we compare the PEA with respect to PathDiP. PathDiP provides core pathway information from major curated pathway database, allowing user to perform pathway enrichment analysis. Now, conversely, from PathDiP, BiP, in addition to PEA, allow to retrieve information enclosed in pathways represented by using the BioPAX format. We download the endometrial cancer mutated gene list that contain 7,000 mutated genes, and we performed pathway enrichment analysis using pathway data available in KEGG and Reactome database. So this is the comparison between the enrichment obtained from Reactome. So BiP enriched 266 pathways, whereas PathDiP 225 pathway. So they have an overlap of 40% among the first 10 enriched pathway. And this pathway include extracellular matrix organization, ion channel transport that are well-known cancer that contribute in endometrial cancer. Instead, in KEGG, we don't have a result because in the available version of KEGG database in BioPAX is an older than that one used from BiP directly from the KEGG database, so there was no overlap at all. To conclude, so BiP is a Java application with which user can simply and quickly perform pathway enrichment analysis, as well retrieve information from BioPAX file. The main advantages of BiP in pathway enrichment is the possibility to perform enrichment from different databases compatible with BioPAX format to produce more informative pathway enrichment result. As future work will regard the possibility to extend BiP in order to make it able to deal with XML, SMBL, and GMTL pathway format. Thank you for your attention.
Thank you, Giuseppe, for your nice presentation. We already have some questions in the chat from Arif. He's asking if do you have plans to this work as a web page or in general a web accessible service as an endpoint to do Java implementation?
At the moment is available on GitHub as a standalone application. And we are not planning, at the moment we don't know if we can share as web services or not, but it is feasible because Java allows easily to transform a standalone application in a web services.
Yeah, maybe it could improve the adoption in the community. Yeah. Oscar instead is congratulating you for the interesting work. He says pathway data themselves are hard to integrate, but connecting pathway databases with other genome databases like gene transcripts, proteins, OMIM, and so on is a great conceptual challenge. Are you planning to enter this integration challenge, and are you using any kind of holistic conceptual model?
Not at all. The only experiment that we did was to try to connect SNP data obtained from the MET microarray data with pathways data, but is still ongoing because it's very difficult due to the huge synonym available for each gene. So it's very complicated to find the correct ID that map on each specific database. So this is the main difficulty at the moment.
So
because it's difficult because you have to prove all the synonym for each gene and see if that particular pathway contains or not. You don't know. So you have to do a semi-automatic approach, and this, I think, difficult to accomplish.
Okay, so I guess Oscar was suggesting that maybe trying to approach it, having in mind a conceptual model may somehow guide you in this challenge. We have a second question from Arif. Is it possible to use this the idea as a tool in a broader view to annotate also the gene ontology?
Yes, this can be done because in Reactome, inside the BioPAX representation of a pathway, there is the annotation with the link to gene ontology. So this is already available in BioPAX. So they do it because they need to understand the cellular component, the function of the particular gene. So this information is available in Reactome. I don't know in the other pathway database, but in Reactome is available.
Okay. Thank you again for your answers, Giuseppe.